Okay, hello everybody. Welcome to day 2 of anatomy e-learning. So this is muscles day 3. Today we have a lot of just like practice and review stuff with a little bit of new information sprinkled in. It's gonna be a great day. I hope that you are all healthy and well. So, agenda, bell work, it's a Google form. I think there are like 3 multiple choice questions and an upload for your IP. Remember to be using your Heron High School email for that. Then we have a lecture overview video, that's this video. And then after that, I want you to watch another 8-minute video about acetylcholine and muscle contraction at the synapse. I'm going to review it, but I want you to hear it a second time from somebody else. The more times you hear it, the more it's going to stick in your head. Fourth thing, practice. So I'm going to have multiple levels of practice up on the haiku page. So you will be able to practice with like a lot of support. So it'll be like, just a few blanks to fill in. And then as you get better at figuring out how this happens, there'll be less and less support. But you can always click back up to that one with a lot of support if you like need a refresher. The deliverable, the thing that I'm going to have you turn in for that is just at the end of your practice session. Please make like a picture or you can make a brief video, or you can write like a short paragraph describing how muscle contraction happens at each level that we cover today. Exit ticket is just a Google form that's like, hey, did everything work? Were you able to access everything? And then your IP for tomorrow is IP number 3. In your packet. It's also linked on Haiku. Okay, so today, before you leave this place, you are going to be able to describe muscle contraction at the sarcomere level, at the filament level, so those actin and myosin filaments with troponin and tropomyosin, and then at the neuromuscular junction or synapse level. That neuromuscular junction, that's our new content for the day. Bell work, 3 multiple choice questions on the Google form, upload IP number 2. I have changed that so you can now upload up to 5 files, so you should be able to get all your IP uploaded. Okay, so review of sarcomere contraction. This is from previous classes. We found out that contraction carried out by myosin and actin is aided by ATP, which is broken down for energy, and calcium, which interacts with troponin and tropomyosin in order to reveal those actin binding sites. We didn't get to make those sliding filament models because that was supposed to happen on Friday when we didn't have school. But refer to your fatigue lab, refer to your notes, refer to videos that we've watched in class to help you out with that. And we're just going to review it right now too. There's a link at the bottom of this slide that I'm going to put on Haiku that is an extra like 4.5-minute video about the cross bridge and myosin attaching to actin that might be helpful for you, so feel free to watch that. I'm not assigning it, but if you feel like you need some extra support. Okay, so I drew some handy dandy pictures this morning. So at the sarcomere level, there we go. At the sarcomere level, remember up here I have actin is this black filament, and then myosin is this like purplish filament, and then that myosin has ADP, adenosine diphosphate, plus a phosphorus attached. So that ADP plus phosphorus give that myosin head just enough energy that it can reach out and grab onto actin, and that reaching out and grabbing on forms what's called a cross bridge. So the ADP and phosphorus get released off of that myosin, and that releases enough energy for what we call the power stroke. And that power stroke is when myosin moves and pulls actin and makes your sarcomere contract. Remember that myosin cannot let go of the actin until a new ATP molecule shows up. And when that new ATP molecule shows up, that is when you get to, that myosin gets to let go, and then that ATP molecule gets hydrolyzed, releases energy, this cycle happens again. But, of course, it's more complicated than that, right? So, we added in at the filament level some other players in the game. So we added in tropomyosin and troponin and calcium, right? So 3 more molecules that you need to know. So in this first picture here, we have the black is still actin. The myosin has turned to orange. I was getting creative colors. The tropomyosin is this green color. There we go. Green color there. And then on that tropomyosin, you see these little dots. That is troponin. Troponin is another molecule that is important here. Remember, troponin loves calcium. There are red dots, and those red dots are the actin binding sites. So that's the only place that myosin is allowed to grab onto actin. Okay, so in our first picture, we have the actin binding sites are blocked by tropomyosin because tropomyosin is there to make sure that your muscles just aren't all contracted all the time. Second picture, calcium shows up. Okay, calcium enters the muscle cell and it binds to troponin because remember, troponin loves calcium. So that troponin binds to calcium and that causes the troponin to change shape. And when the troponin changes shape, it moves the tropomyosin and it reveals those actin binding sites. So the place where myosin is allowed to grab onto actin is revealed. That is when the ADP and phosphorus allow that myosin to reach out and grab onto those actin binding sites. And then you kick off that ADP and phosphorus, and that is when movement actually occurs. Okay, so that should have all been review. The next slide is not review. So this is at the synapse level. So if you're wondering, where does the calcium come from? That is essentially what we're going to answer right now. So a synapse is where you have a nerve getting close to your tissue that it's going to try to send a message to. Remember, your nervous tissue are made out of neurons, and neurons, you have the cell body, which has the nucleus in it, and then you have the dendrites, which are the little spiky hairs close to the cell body that are the receiver of messages, and then you have the axon, and the axon is that big long tail that sends messages, right? So in this case, we have the axon terminal right here. So that just means the end of the axon. And that axon terminal receives a signal, all the way down the axon. And what happens is it starts to allow sodium, which is this Na+, and calcium, which is Ca2+, to come into the axon terminal. So these things start entering the axon terminal. That calcium will attach to acetylcholine. So acetylcholine is a neurotransmitter, which just means it's transmitting a signal. So this acetylcholine is like the messenger that's going to go tell the muscle, hey, it's time to contract. Okay, so calcium attaches to that acetylcholine and it moves the calcium out, or it moves the acetylcholine out into what's called the synaptic cleft. The synaptic cleft is just the space between the axon terminal and the muscle. So this looks like it's a big gap. It's not a big gap. It's very, very close together, okay? So that acetylcholine enters the synaptic cleft, and then it attaches to this sodium receptor on the muscle fiber. So muscle fiber equals muscle cell, right? So the acetylcholine will come in, it will attach to that sodium receptor, and that says, hey, gates are open, time to let sodium in. So sodium, notice it's only a +1, right? So it has a lesser charge than calcium has. So sodium starts coming in to the muscle fiber, and what that does is depolarize the membrane just enough that it will that muscle fiber will start letting calcium in. So sodium comes in, depolarizes the membrane, and then calcium starts coming in to the muscle fiber, okay? It takes a lot of calcium to make your muscles contract, right? Because it's binding to troponin all along that muscle fiber. And as it does that, it's moving the troponin, which then moves the tropomyosin, which will then allow the actin to bind to the myosin, or myosin to bind to the actin. So you have to have a lot of calcium to do this and to repeat your muscle movements. Okay, so you do have calcium coming in, but you also have that calcium binding to what is called the sarcoplasmic reticulum, which is here. So that sarcoplasmic reticulum, when calcium binds to it, it's a storage place for calcium. So it will dump all of its calcium once calcium binds to it, and that is called calcium-induced calcium release. And that just like we're flooding the muscle fiber with calcium. That way you have enough. Calcium to make the muscle contract. That calcium can also go to other cells in order to make them contract too, because you don't just have one muscle cell contracting, you have many. This slide, this is going to be put up on Haiku. This is just key players in our saga, so it's just like a kind of like a brief rundown of all of the different molecules and like where they take like action. Okay, lab results. Hopefully you saw some trends that you started to fatigue towards the end of the trials. Some people will fatigue towards the end of the trials and then they'll like have another boost of energy, so their graph will look more like this. So think about how you can modify this to better demonstrate fatigue or to measure for isometric. Ignore that second part. Okay, so next thing is the upcoming video. It looks a lot like what we just did. It just goes more in depth. So take notes on it. And then your practice, there will be a link on Google Forms. And there's going to be practice with varied layers of scaffolding. So like the first set of practice is going to be like very, very scaffolded with a lot of supports to get you like just get the ball rolling. And then as you start to feel comfortable with that, try one that has less scaffolding just to try to get it. So it's like, you know, riding a bike with training wheels. So try to take those training wheels off and really get to describing this process like on your own. Okay. And then at the end of the practice, draw a picture, write a paragraph, or make a short video to describe the process of muscle contraction and upload it to the Google form. Cool. As always, email me with questions. Muscle IP number 3 is due next class. There's a muscle reading and coloring that goes with that. So like you don't have to send me the picture of your coloring, but do IP number 3. Okay, IP number 3 due next class. As always, I have office hours every day, 1 to 2 p.m. Chester made an appearance today. So stop by. It's a good time. I'd love to see you. I hope that you are well. I will see you on Friday.